Hello everyone and welcome. Thank you for joining us for today's webinar, Best Practices for iNaturalist Photography. My name is Dr. Ashley Burke and I'm the Marketing and Communications Officer for the Entomological Society of Southern Africa. Today's webinar will be presented by Jermaine Mugai, ESSA's Early Career Entomologist Representative, together with Dr. Cassandra Barker, a postdoc research fellow based at the KwaZulu-Natal Museum. Whether you're an experienced photographer, an entomologist, or simply someone who enjoys observing insects and other arthropods, we hope today's session will provide practical tips to help you capture better photographs for iNaturalist and contribute valuable biodiversity records. Before we begin, I'd like to take a few moments to introduce ESSA and share some opportunities available to our members. The Entomological Society of Southern Africa is a vibrant community that brings together professional entomologists, students, researchers, educators, and insect enthusiasts from across the region. By becoming a member of the ESSA, you'll join a supportive network of people who share a passion for insects and arthropods, with opportunities to connect, collaborate, and grow throughout your career. Members can apply for a range of grants and awards, including the Rostrum Photography Prize, the Young Entomologist Essay Prize, and the Young Entomologist Travel Grant, all designed to recognize and support emerging talent within our field. ESSA members are also encouraged to publish their research in our journal, African Entomology, with discounted article processing charges available to members. If you'd like to become part of our community, simply scan the QR code on your screen to visit our website and learn more about membership. We'd love to welcome you to the ESSA. To stay up to date with ESSA news, upcoming events, webinars, conferences, and opportunities, visit our website by scanning the QR code shown on your screen. If you have any questions about membership or the society, you're welcome to contact our administrator using the email address displayed here. We also love hearing from our members and the wider entomology community. If you have entomology-related news, upcoming events, achievements, student successes, or other announcements you'd like us to share through our communication channels, please do get in touch. We're always happy to help showcase the exciting work happening across Southern Africa. Before we begin today's webinar, we'd also like to highlight an exciting event on the horizon. In July 2028, South Africa will once again host the International Congress of Entomology, the world's largest gathering of entomologists. After 20 years, this prestigious congress is returning to South Africa and will bring together thousands of researchers, students, and industry partners from around the globe. We hope you'll join us in Cape Town from the 17th to the 21st of July in 2028 for this landmark event. You can find out more and register your interest by visiting ice2028.com or scanning the QR code on screen now. And now, without further ado, I'll hand over to Jermaine, who will take us through today's webinar. Enjoy the session.
Moving on to today's topic, Dr. Cassandra is going to guide us on how to improve our iNaturalist observations. Just to introduce the platform, iNaturalist is an incredible global biodiversity networking platform, one of those projects that started very small and now it is global. It is connecting nature lovers all over the world, and it is a user-friendly platform. You can get guidelines online on how to engage on the platform. There are videos, video tutorials that shows you how to use the platform. They also have a mobile app that you can use while you are out in the field, and you can capture your specimens and upload on the app. You can explore, upload your findings, identify, track, and also join groups or find projects on the platform and get to work with other people. It is heavily used right here in South Africa. It helps us to map our local biodiversity. You can see the numbers, you can see the number of species, it's a lot, and identifiers and observers. And currently, there's over 200 million observations across the globe. So it is an active platform. It is also a very helpful platform. For an example, in the newsletter, in 2024, a mole species was spotted by a Swedish tourist in New Zealand, and this species was considered extinct for over 50 years, and now it's been spotted and people are documenting it, and it has since been documented on iNaturalist almost 20 times. So this platform is a platform that can also help us to cover the gaps in science. So it's very, very useful to be on the platform and see what other people are capturing, and you can maybe see if you can cover the gaps in your research. And with that, I will now hand things over to Dr. Cassandra. A warm welcome to everyone, and over to you, Dr. Cassandra.
So yeah, Charmaine has kind of covered how this is a community-driven platform, that it really depends on how people interact with iNaturalist, and that really boosts the value in the observations that we get. And she did mention an example from New Zealand, but to bring it home into a local example, I've been working on Bombillidae or bee flies for the last few years. And my special interest is a group of bee flies called Mariobesinae. They're abundant pollinators on the Maquilan daisies. And there is one genus I've just really struggled to find. It's got very sporadic records in museums. Until this year, when there was an iNaturalist observation uploaded, and this specimen that we can see here was found just outside Prince Albert. After a few messages back and forth between myself and the observer, and we got landowner permission, I was able to go down and collect a fresh specimen. And this one specimen we collected is going to contribute to quite a few scientific articles in the future. So it really illustrates how valuable an observation can be to ecologists, taxonomists, evolutionary scientists. It doesn't just go into a void and get ignored. But enough about me. Let's get on to why you're actually here. And I'd like to start with what not to do. Some of these points may seem obvious, but they are worth mentioning. Also, this is not to embarrass anyone. It's really just to manage expectations when participating with iNaturalist. I ask that if you do see anyone on the platform making these mistakes, either direct them to this webinar or give a gentle suggestion. All right, so the first point regards blurry photographs. Some people believe that a critter, arthropod, insect is quite easy to identify from a picture, and the quality of the image isn't that big of a deal. However, a blurry photograph is going to obscure the traits that would help identification. Sometimes a blurry photo may result in an identification, but that really depends on how active the experts are on iNaturalist, and also how charismatic or well-known the group is. So we have the example of the deathhead's hawkmoth, which is really easy to see, really easy to identify. And so when you have almost like a gray blob uploaded, it will get to a species identification. However, something like a bee fly or bombillid, the best you're going to get is a family level identification, which can be quite frustrating to some people when you've gone out into the field and you've put in effort to try and take these images. The next point I want to make is squashed insects or arthropods. Again, it's easier to assume that these critters are easy to identify, but in the case of squashed animals, the characters that would be needed to provide an identification are damaged or missing because that they're squashed. And so it's basically impossible to provide a reasonable identification. Even your top experts that work on spiders might just tell you, this is a spider. That's the best you can say. And then the other example I'd like to touch on is images of specimens that are just too far away. And I understand that sometimes you want to get an image before it flies away or scuttles off, and we'll touch on improving this in a moment. But really, with this kind of focus being so far away, the best you'll get is a family level identification. It'll be quite a stretch to get to a species ID. And then the stylized images. This is not a common upload, but it is worth mentioning. Stylized photography has its place, especially when it comes to illustrating the beauty of all creatures bright and beautiful. However, they do tend to not focus on diagnostic characters. It's more about how striking the subject of the photo is. If you want to include your stylized photos, by means do, but try to include other images that would be from different angles, and that'll provide more information on the animal that you're looking at. Also, if you're interested in sharing your artistic photography that focuses on animals, there are various active groups on Facebook or subreddits, and these kind of stylized images would really be greatly appreciated on those platforms. But enough with the negativity. Let's talk about what you want to do. So you found a critter and you want to take images for it. You want to upload to iNaturalist. The best thing to do is to take as many photos as you possibly can of the individual at different angles. Not only will this help overcome some of the obstacles of like a blurry image or images too far away, but you'll be capturing many different characters that will help with identification. And you might even capture behavior that could be used in a description, revision, or short note, something that might stand out to someone who works on these animals that you wouldn't realize would be something interesting. I do understand that iNaturalist supports videos now. However, not everyone can take a video of the critters that they're looking at. And then again, going back to the example of images of individuals being too far away in your image, if you are including multiple images, you can take a photo from far away, and as you get closer, you're taking another image, and that way you'll at least have a few that aren't blurry, aren't too far away before whatever you're looking at scuttles off. And then when it comes to the angles, if you can only get a single image, the best option is to do a dorsal view with a slight angle. And this really provides the most that you'll see of a critter, and it'll provide the most information for identification. I have been asked that if you are interested in finding scorpions at night, to please make use of a UV light. This is a scorpion under UV. They come up beautifully. It can be a bit eerie the first time you do it because they just stand out against the darkness. And Ian Engelbrecht, who works on scorpions and spiders, has mentioned that these images of specimens can be used to identify them. So please, if you're going to look for scorpions, take a UV light. They're quite inexpensive and it can make your searching and iNaturalist uploads a bit more fun. But yeah, you'd want to do a dorsal view or a view at dorsal with an angle to make sure that you're capturing as much of the specimen as possible. And then to give some trait-specific information, any organisms or animals with wings, you want to make sure that you're including the wings in your pictures. These pictures should be in focus with the wings prominent. And that will really give information on the group, so the family, all the way down to genus. It'll also make it easier to identify your specimens. So for example, with this specimen, because I can see the wings very distinctly, I know that this is a fly that mimics wasps, as it only has 2 wings. Wasps and bees have 4 wings. Also, flies have these halteres. And I'd be able to get it a bit further down to species level based on the structure of the wings. So the vein pattern, the coloration on the wings, this is important information. And then if you see specimens with amazing antennae, or just you can get images of the antennae in general, include that in your posts, especially in cases where the antennae are quite elaborate. It's very likely that the ornamentation of the antennae is important to determine which species it is. And this advice could apply to pincers, mouthparts, or even the end of abdomens in the case of dragonflies and damselflies. You could think to yourself, what about this creature lets me know that it is that creature? So if you're looking at a beetle, a fly, a butterfly, how do you know that this is the animal that I'm looking at? And then that'll help you just focus on which traits to take a photo of and upload to your iNaturalist observation. And this is another point, I would say, the environmental images. I would say that this aspect of the observation we don't see often, mostly because a lot of people think that where we found or saw a critter is not as important as just taking a photograph of it. However, if you could include an image of where you found the critter, it could provide information like the soil type, if you found the creature in an open field or in a forest. Some critters are very specific on where they like to live. And so this kind of information, it can broaden our understanding of the ecology or life cycle of the individual. And if you're someone who doesn't respond to direct messages from other people on iNaturalist, these kind of images will help them go find it if it is of importance and if it's a part of a ongoing research study. And if you see a organism interacting with a plant, whole, or anything that gives an insight into that individual's diet or living conditions, include that in your observation. For example, we have this image of a nemestrinid, and I would say, and this plant isn't a part of the original upload. However, I would recommend adding this information if you could say in your notes that you saw it pollinating this kind of plant. And you could even upload the image of the critter in one post and then the plant in another post and add the link to either observation in the notes. That's especially useful if you saw a pollinator on plant that could only get a photo once it landed on the ground away from the plant it was visiting. So you could essentially put up 2 posts and cross-link them. And that'll be a great way of linking up your observations and providing more information on what you've been seeing when you're out in nature. And so let's expand on the observation notes. This is your opportunity to provide information that you couldn't share in a photograph. Like I've said, this could be a link to a plant you saw, the individual pollinating, or the hole you saw the critter coming out of. It could also be behavior you observed. For example, I once spent some time watching an individual of bee fly that is on the slide that was fluttering its wings and was very obviously performing a courtship ritual. And this kind of information could be of value to someone who wants to understand the wing patterns on various insects and see how that's a part of a communication strategy. So this is a way to elevate your observations on iNaturalist and really expand it towards natural sciences. And the last item I want to talk about is when it comes to taking photos. So smartphones today have better and better cameras, and it's possible to order quite inexpensive lens attachments that you can easily clip on when you're out enjoying nature. So you don't have to have a DSLR camera to upload onto iNaturalist. Start with your phone, and if you enjoy uploading to iNaturalist, once you're more comfortable uploading to iNaturalist, you can buy one of these camera attachments off Takealot or Temu and use it when you go out into the field. I really hope that these points we've provided has been insightful and that you have a better understanding of what to do when you are seeing a creature out in the wild and you want to upload to iNaturalist. So I'd like to thank all the people who've provided some information for this presentation. It wouldn't have been possible without them. So thank you to all of them. And we will open the floor to questions. Thanks.
Thank you, Dr. Cassandra. As we are waiting, I just want to remind everyone we are having our AG meeting next week. Please join us. You have the link in your inboxes. Please visit your inboxes. You will find the link. Yes, we may have questions now.
Hello, I just raised my hand. Steve Woodall here from NEPSOC.
Hi, Steve.
Hi, how's it? Hi, everybody. I spend a lot of time identifying butterflies mainly and some moths on iNaturalist. And the one thing that really, really gets me is when you get somebody who takes 20 photographs of different butterflies and puts them all on one post. We need to make it absolutely clear when they log into iNaturalist, it's one organism but upload, no more. Because it just makes, it's so frustrating. You see all these beautiful images, you can't identify them. You send them a message saying, please split them. Sometimes they do. I've got 3 or 4 hanging around on my feed that have not changed for 5 years. So, yeah, that's my 2 pence. Make life easier for us.
Yeah, that's a really good point. And sometimes I'd recommend that people can double post. So I've seen it where there've been Coleoptera and Diptera in the same picture because they're interacting with the same flower. And so you could probably double post where you're asking for the identification of the beetle in one post and then the identification of the fly in another post. It's quite a common post I see on iNaturalist, but you are right to expect numerous individuals to be identified from one image is very frustrating.
Dr. Bakker, we have some questions in the chat. Shall I read them out?
Yes, please.
So there was a comment earlier from Tony Rebello. I'm just going to read this out. It was about when you mentioned the blurry photos. And his comment was that the blurry photos may be difficult for specialists, but many people want an identification, and an ID of a bee fly may be all that they need. So the site is for everybody, so these should rather be viewed as an opportunity to hook and educate the users to taking more and better observations. I don't know if you want to comment on that, but my feeling is always, we always want to educate. We want people to be interested and involved in entomology, but we do also want to help people improve their observations, which is the point of this webinar. Don't know if you want to comment further on that.
Yeah, so Tony, thank you. Tony is so incredibly involved in iNaturalist, and he is very right. This is a community-driven project. And yes, the point of this webinar is to get a better idea of what would help if you wanted to improve your observations on iNaturalist. Yeah, if you're going into an area where there have been no identifications, a blurry photo is better than no photo. However, to manage an expectation, to expect a person who does identifications at a species level to provide a species level identification of a blurry individual is not a good expectation. So by all means, if you are in an area and the only thing you have is blurry photo, you can upload it. Just it may only stop at a certain point and not go further. So that the don'ts were more to manage expectations, not to embarrass anybody.
Thanks very much. There is also a link that Tony provided in the chat to linking observations with such as pollinators with certain projects. So that has been linked in there as well. Right, may I go to Bruce and Alice's question, which is, I think, directed to Steve Woodhall, which is, how do you get butterflies to sit still long enough to get a decent photo? And I think maybe if Steve can comment, but also in general, when insects are flying or moving, what's the sort of best approach do you recommend?
Okay, I actually answered this and said patience and stealth and a long lens, because generally, certainly with butterflies, if you keep still for long enough and if you don't make any sudden movements, they eventually they should get used to your presence and they'll sit still. But they don't all do that. Some butterflies only sit with their wings closed, so you need to get a picture of the underside at least. But with some of the new DSLRs, not DSLR, but new mirrorless cameras, you can actually put them in machine gun mode, like I did today, and get photographs of their wings opening and closing in flight, which is unbelievably good. But yeah, it's a long lens is a really good thing to use, and just gentle movements. Don't chase after them. Try and avoid stepping on twigs and that sort of thing and breaking them because they can hear. And yeah, that's my secret. That's how I get so many good close-up photographs. I've got a 1:400 zoom lens with a 1.4 times converter and an R7, and it allows you to get close to most butterflies, even when they're sitting high in the canopy. I can still do it.
Wonderful. Thank you so much. That's really interesting. Cassandra, do you want to add anything, or we can move on?
We can move on. It's great.
Perfect. So there is a question that also Tony has also addressed in the comments, but just for the sake of everybody else. So the question is there a way of hiding location on iNaturalist? So specifically in the cases where you're dealing with threatened species or very sensitive species. The response is that it is automatic for sensitive species on iNaturalist, for example, rhinos and succulents, but not all threatened species need to be obscured, only if the locality further threatens the species. Obscuring species threatened by aliens or climate change or agriculture is silly, even if they are critically endangered. If you are trespassing or think a species could be compromised if the locality is known, then choose the obscure option. But note, no one except you and the species you trust specifically can see this locality. Is there anything else you'd like to add to that?
I do think that it's worth noting that poaching is a very serious occurrence in South Africa. So if you are worried about baboon spiders or scorpions, some insects, I'm not too sure if they're poached heavily, but if there is something that you are worried might be poached based on your iNaturalist observation, by all means obscure it. But yes, as Tony says, obscuring agricultural crops would be a bit silly. But if you are worried about someone doing something heinous with your observation, you're welcome to obscure it.
Thanks very much for that. All right, and the next question is it possible to identify thrips species with iNaturalist?
I do not work on thrips, so I can't say for sure based on that group. However, I can take it from the point of view of Diptera, and it is incredibly difficult to identify Diptera to species level based on a photograph, usually because there are characters that you cannot see on a photo. It'll be underneath or it'll be very small. So there are some taxa that we can identify to species level just because it may be one species in a genus, and so the identification is very obvious, or it's something incredibly charismatic, and so it's easier to identify. The answer will kind of depend on which group you're working on. However, it will most likely be very difficult to get to a species level identification just from a photograph.
Yeah, I was just going to say that, so Steve here again, I know that's the case with dung beetles, for instance. I mean, Vaughan yesterday said to me many times, you can only get to the genus, and even then it's sometimes difficult. And we've got some butterflies like the genus Leptotes, where you literally have to dissect the genitalia to tell them apart. So I think what we did there, Tony created a complex, so we can just say it's complex Leptotes pluritax. And so we just said to people, sorry, it can be one of 4 species. That's as far as we can go. And the same thing goes with a couple of other groups like the subgenus Afrogegonese, for instance, where it's impossible to tell them apart from the underside. I just take it to the subgenus, and I stop the community taxon there and don't move it any further. That's one thing you can do, by the way, is if 2 people have agreed on it at a higher level, a higher taxonomic level, you can actually halt the process at that level and say we can't go any further than that.
Yes.
Hi, I would just like to add here, it's on the topic, Sun here, also from LabSoc. The same goes for caterpillars in general. There are, of course, common species that, we can identify to species level just from photos, but more often than not, caterpillars cannot be identified to species level with certainty from just a photo. So in that case, we always advise for people to rear them if at all possible. We do have dedicated official ESSA projects that collect all the rearing data there. So that's always a consideration. Caterpillars always best reared to ensure you get correct species level ID.
And there's a project you can use to link them all as well, by the way, which sub-runs for LabSoc, which is a great way of doing it. I see Alessandro said, please, I need assistance with these flips. Ultimately, you may find that a photograph's not going to help you. Sometimes you do need to put things under a microscope. Sometimes you do need to dissect them. But I think with iNaturalist, you actually can put those observations on as well, can't you? You can put a mounted specimen, you can put a microscope slide on iNaturalist. Just ask Tony, is that allowed?
Yeah, I have seen it happen, and I've seen people put up their pinned specimens. I think the only issue there is your correct locality.
Exactly.
Because I would assume that if someone's doing it from their cell phone, they collected the specimen, say, out at Thomas Baines, took it back to their lab and pinned it, took a photo, and then the locality is whichever lab they were in. That's where the issue would come in.
We've seen that. And we take them to task about it as well.
Yeah.
Take a GPS with you, photograph where you saw it, where you caught it.
So what I like to do is that if I do get to a species level identification, just because I'm so incredibly comfortable with the group in that we're looking at, I will try to add characters under the identification, just as a tool for teaching. For Alessane, what you can do is you are welcome to email the society, and we'll try to help you get in touch with someone who works on thrips. And then we can try and help you with your PhD. Taxonomy is quite hard, but there are resources. So you're welcome to email the society, and we'll try help you get your specimens identified. All right, I've seen there's some other questions. So Ashley, are there any other questions in the chat we should highlight before we get to the raised hands?
Well, I think let's go to the hands.
This thing about difficult groups in LabSoc, we're trying to, through iNaturalist, we've created groups in the various countries, and what we're trying to do is to contact whoever is the world's top authority on that particular family of moths, particularly moths, and get them to join iNaturalist so we can tag them. Because we haven't mentioned that you can tag, if the person is actually on iNaturalist and they've got a username, you can tag them in the upload and they'll see it. That can be a tremendous help. We do that a lot.
Yes. My only concern is that we don't want experts to be bombarded on iNaturalist. So this is a community driven. Yes, it's polite to ask them.
Can I tag you? Yeah, that's what I do. Yeah.
Perfect. All right, let's go to the hands. So Shuna, I hope I've said that right.
Hi, Cassandra, it's Sinead, but it doesn't matter. Nobody
Sinead,
can
sorry.
figure out my name from just looking at it. No worries.
Sorry.
Before I get to what I wanted to say, I just wanted to add from ESSA's side, and Charlene should be on the call so she can confirm with me. But I do believe that ESSA as well does have a team of people that can consult. I don't know if there is per se a THRIP specialist on the list of people that are there, but that is also another option if you are stuck, is to email the Entomological Society, because there are people who are who have been put together that have been tagged to identify things, and hopefully they might also know someone who can assist. So that's the first thing. So the second thing from my side is I'm not an expert like the people clearly who's here. So I'm approaching this question as, say, I'm a casual person. I've taken a picture. I want to know just generally what it is. There are different types of groups. So you've got iNaturalist. I've seen different groups on Facebook, etc. So could you give an idea of the utility of the different type of groups? Like if you asking iNaturalist, what's the utility of that versus asking a group on social media, bearing in mind that even from my side, not being an expert, I have seen some truly heinous misidentifications on the butterflies, bees and things group on Facebook, where some of the bitey flies have been very highly mis-ID'd. But so basically what I want to know is for each of the different type of places that you can go and get IDs, how much of a level is what level of expertise are associated with each of these different groups where I can go and identify things. I hope I'm making sense.
Yes, that is a really brilliant question. Thank you so much. So I will first talk about the Facebook groups. These tend to be people's best guesses, and compared to so how it works in iNaturalist is you'll upload your image, your observation, and then you might say, okay, I think this is a beetle. And then that a lot of people who frequent iNaturalist would be looking up beetles that are unidentified, and they might say, okay, I know this group, it is this genus. And then someone else will agree with them. And then you have another person agree with them. So there isn't much discussion. The discussion might come in when someone disagrees and they'll say, oh, but it's missing this character or that character. Compared to a Facebook group, you might have someone saying, oh, lovely picture, or where did you find this? There isn't really a drive to give an identification. The Facebook groups can tend to be more conversational. The other thing, which is how incredibly useful iNaturalist is that the raw data can be scraped for publications. So you could say that you are interested in creating a species checklist for a reserve, and you download all the iNaturalist observations for that reserve. And you do a species analysis to look at diversity, to look at where most observations are happening. These are publications that are coming out. So we're seeing iNaturalist being used as I want to call it a data repository almost, whereas the Facebook groups are more community conversational. I've never seen anyone actually use the data from the Facebook groups. So if you are casual and you want to just contribute to our understanding of species diversity, I would definitely recommend iNaturalist above a Facebook group.
Okay,
I hope that's answered everything.
Yeah, because like I said, even of my limited understanding, I've seen some terribly misidentified things, even in things that with some mosquitoes and horseflies and things of the sort. So you get some very pretty pictures, but the IDs are a little bit on the sus side. So okay. Thank you.
I've got mixed feelings about Facebook.
Yes.
Some of you here may realise I've got a bit of a high profile, and I'm constantly being bombarded with it says IDPLZ or just ID question mark, which is bloody rude, and you know, and which I've got to the point now where I said, listen, don't be so rude. Say please, number one, and number two,
Please
I'll
spell it out.
identify. Here it is. This is what it is. Now please go to this, and I paste the iNaturalist tag into the comments on the Facebook. Please sign up for this, put it on iNaturalist, and then tag @Steve Woodall in the observation. And but I'm not going to do it for free on Facebook anymore. I'm sorry, you know. and that usually they walk away in a huff and don't come back, which is fine, or they start. We've got a lot of new people have started doing iNaturalist because of that, which is great, you know.
Cool, thanks.
All right, thank you. Melissa, I see your hand is up.
Yes, sorry. In Port Elizabeth.
I'm sorry, Melissa, I'm struggling to hear you.
Hi, is this better?
Yes, thank you.
Hi, sorry. I'm Melissa. I work for Citrus Research International in the Port Elizabeth office. I am in integrated pest management. I have a comment on the thrips question that was asked a bit earlier. We have I have a colleague who is doing his PhD on thrips, and I know that when he identifies his thrips, he can't do it without a microscope, because you need to use the patterns on the wings for a good identification, as well as some other other characteristics as well. So it's very difficult to get identification on thrips just from a photograph.
All right, thank you so much.
No.
It's really great to get this expert information and advice. It's the great thing about this webinar.
Interesting. I really enjoyed the talk. It was very, very interesting, and I will definitely use it more often. I haven't used it in a long time. So yeah, thank you so much for it.
You're welcome. All right, Sun, and then I think we can wrap up.
Sorry, that stayed up from earlier. I'm good. Thank you.
All right. Are there any more questions? No. All right. So if there's any part of the webinar you might have missed, this has been recorded and it will be uploaded on YouTube probably within the week. So last time, I'd like to thank everyone who took time to be a part of this webinar. We've really enjoyed presenting it, and I hope you got something out of it, even if it's some funny stories or anecdotes. Thank you all again for coming, and hope to see you soon, or see your observations uploaded. Right, thanks everyone.
Yeah, thanks everybody. It was good fun.
Thanks for the great presentation, Cassandra.
Thank you.
Bye.
Thank you for joining us.
Thank you.
Thank you. Thank you.
Thank you. Bye.
Thank you. Bye-bye.